Okay, we are going to look at taking a naive but mature T cell and getting it to become activated. And this is going to happen through interaction with specific antigen. Now, the way that we're going to talk about this, it tends to be kind of an easy way to look at it, where, there's a break in the skin and the pathogen gets in that method. Please know that this is not the most common way that pathogens get into the body. Most often, they're going to get in through existing openings, pores in the skin, but a lot of them through like the respiratory tract, the oral and respiratory tract. So just know that even though we're talking about a break in the skin, and like here we have the popliteal lymph node being the draining lymph node, most of the time this is going to be happening in the upper respiratory tract. Okay, so but it's all the same, the process is all the same. And then we're looking at lymph nodes in a lot of these examples, but know that this is going to be very much the same as if it was like a mucosal associated lymphoid tissue. Any secondary lymphoid tissue is really what we're looking at, but we tend to use the lymph node as our example, just so that you are aware of that. And in any case, a pathogen has to gain access to the internal tissue, get through the epithelial layer and make it down into the tissue where those resident dendritic cells are living. We haven't talked a lot about dendritic cells, so let's spend some time and look at them with just a little bit more detail. So there are a few different types of dendritic cells. The ones that we're concerned about in this chapter are myeloid. They're from the myeloid lineage. So they come from hematopoiesis in the bone marrow, just like what we saw with all the other white blood cells and red blood cells and platelets. And dendritic cells are absolutely central to the activation of T cells. They are the cells that are going to be resident in the tissue right below the epithelial layer, resident anywhere where pathogens can get, that will capture antigens, internalize antigens, process them, and then carry them to the secondary lymphoid tissues to interact with naive mature T cells. And so they are the best of the best antigen presenting cells. Macrophages and B cells are going to have a small role in antigen presentation, but nothing like dendritic cells. Dendritic cells are going to be pretty much the only cell that is able to activate a naive T cell. Pretty much. Macrophages can do it a little bit, but dendritic cells truly are the hero here. They will, out in the tissues, take up pathogen, then they'll process it and present it. But once they get into the secondary lymphoid tissue and they're doing their presentation work, they're not going to be taking up any more pathogen. They've already picked up what they're going to pick up. They've migrated to the secondary lymphoid tissue, whether that's a draining lymph node or some mucosal associated patch of cells. They will then interact with T cells and upregulate their expression of MHC class I so that they can present to a lot of different T cells at the same time. So to activate a mature, naive CD4 positive T cell, so this is a T cell that has gone through complete development, positive selection, negative selection, and is deemed safe to be out in circulation. The dendritic cell is going to have to process any antigen that's been taken up into tiny little peptides, attach those peptides to an MHC class II molecule, and then display them in conjunction with that MHC class II molecule. Because recall that CD4 is the co-receptor. So the antigen will be bound by the T cell receptor, and then MHC class II is bound by CD4. Receptor-mediated endocytosis is the process that bacteria and maybe some floating around viral particles are picked up from the extracellular fluid by the dendritic cells. And so these are considered somewhat extracellular pathogen. And so that's why they're going to be presented on MHC class II. Macropinocytosis is the process of bringing in fluid. But regardless, MHC class II will present exogenous antigen. But we do know that there is a chance that a dendritic cell could by itself be infected by a virus, and it would be endogenous, or the virus can be picked up by endocytosis and cross-presented, even though it's brought up through the through an endosome, it's still recognized as an internal or intracellular pathogen. So then these situations, the pathogen would be processed by a proteasome and added to MHC class 1, right? Because internal pathogen like viruses are presented on MHC class 1 to CD8 positive T cells. So kind of review, but it's important to know that dendritic cells are the big antigen presenting cells here, and they will present with both class 1 and class 2 MHC molecules to activate both CD4 and CD8. Knowing that exogenous pathogens like bacteria and other potentially viruses that are just viroid particles out there on CD4 or MHC class 2 for CD4, and then internal pathogens like viruses for sure on on MHC class 1 to CD8 positive T cells. Another receptor that we haven't talked a lot about in a while are toll-like receptors. Toll-like receptors are very important for the uptake of these pathogens, whether it's a bacteria or a viral cell. And so toll-like receptors are abundant on dendritic cells, making them very sensitive to taking a pathogen. So you have a dendritic cell that's out of the tissue. It ends up taking in pathogen, whether it's, well, in this case, it's bacteria is the example that's showing. As it takes it up, it's processing it. The dendritic cell is moving to the secondary lymphoid tissue. Here we have a picture of the draining lymph node. We use the term draining lymph node to describe any lymph node that is closest to the site of infection. So this could be, we have lymph nodes throughout the body, throughout the lymphatic system, or any of those mucosal associated tissues. They'll enter that secondary lymphoid tissue through high endothelial venules and interact in this area called the T cell zone. It's a rich area full of T cells that allow for dendritic cells to come into contact with many T cells. And hopefully the T cell that has the specific antigen receptor for that process antigen will come into contact with it. If it doesn't, then the T cell, let's see here. T cells will also then enter, okay, and then that mixing will happen, okay. So this microanatomy then of the lymph node or of the secondary lymphoid tissue does account or does allow for this mixing of T cells and dendritic cells to happen in this T cell area or the T cell zone. It's going to be towards the outer cortex area of the lymph nodes. And yeah, it's just a lot of transient interactions. T cells have their receptors. They're looking for their specific antigen, and they're just looking. And there's dendritic cells that are presenting and showing. And maybe, just maybe, a specific T cell will find its specific antigen. And if that happens, then there's going to be a complex or a synapse that's formed. Let's see here, and it's stayed for, it's retained, it's selected to stay then in the secondary lymphoid tissue. This doesn't happen very often because chance has a lot to do with it. Physics, of course, has a lot to do with it. And so it may happen, it may not happen. And if it doesn't happen, then that T cell just leaves and moves on to the next lymph node and tries again and interacts. And it just keeps going in and out of secondary lymphoid tissues looking for antigen that is specific for its T cell receptor. So most of the time, T cell's not going to find it. And it just keeps circulating. And then these circulating naive T cells will just continue to do this for the rest of their lives. And they can live a very long time. They'll just self-replicate. They'll just divide. When they start to get old, they're like, okay, it's time to divide and make new cells. But eventually, every T cell will make its way through all the secondary lymphoid tissues and, just continue circulation. This just constantly goes on throughout the individual's life. This process that T cells are able to find secondary lymphoid tissue is called homing. So T cells will home to secondary lymphoid tissues based on chemokines. We mentioned a few chemokines previously. One of the big ones was CXCL8, which brought in neutrophils into an area of a bacterial infection. Well, CCL21 and CCL19 are big chemokines that are secreted by the stromal cells and dendritic cells within the secondary lymphoid tissue, and those will act as like addressins to bring in T cells. And so the selectin, then the CCR7, that is expressed by a T cell that will interact with those chemokines and be pulled into the secondary lymphoid tissue. So it is, the process is called homing. It's not like that name isn't specific just to T cells, but it is the process that T cells use. But it's all about chemokines driving traffic. So as the T cell, again, mature, has a functional T cell receptor, naive, it has not been exposed to specific antigen yet. But as it makes its way through all of the secondary lymphoid tissues and it's just packed in there through that T cell zone of the cortex, they're just going to test and sample and check and what's this one? What's this one? What's this one? Will I find my specific antigen this time around? You just never know, right? And so these interactions are going to allow for the T cell to kind of just assess, okay, you have MHC class 2, I'm CD4, what does your antigen look like? And then if it doesn't match, well, then the T cell goes on as the next one. When there is a match though, and that T cell recognizes antigen, the co-receptor recognizes MHC, that will start signaling to the T cell to stay put, stay attached, and prepare for activation. It'll be a really exciting time for that T cell. This interaction between the T cell and the dendritic cell is going to be stabilized, and they end up becoming what's called a cognate pair, or a conjugate. This can last for several days, where the T cell and the dendritic cell continue to send signals back and forth, but it is a pretty strong connection of those cells. Once then that T cell has that connection to the dendritic cell in the secondary lymphoid tissue, it's essentially kind of trapped there. It's becoming activated, and the dendritic cell is sending the T cell messages to start replicating. Go ahead, T cell, start proliferating, because I provided you with antigen that is causing an infection, and you have the specific T cell receptor for this antigen. We need you. We need your cytokines to drive the immune response. And so under the influence of that dendritic cell, the T cell is going to replicate several times over. And they're all going to stay attached to that dendritic cell. And it's kind of like a nursery. This dendritic cell takes care of all, it has all these dendrites, right? And so kind of like that picture shows, and it has all these little spindle dendrites that are going to like take care and surround, protect and connect with these T cells so that they can propagate and they can mature and they can start getting ready to create the cytokines that they need to have to go out and drive the immune response. But that would be too easy if that was just the case. Simulation actually takes more than just the dendritic cell interacting with the T cell through the T cell receptor and MHC. That signal is required. So the CD4 positive T cell in this case has to recognize MHC class 2. The T cell receptor antigen specificity has to recognize the antigen, but that's just the first signal. There needs to be an additional signal, or what we call a co-stimulatory signal, that is also sent to the naive T cell to get it fully activated. And this second signal is delivered when B7 binds to CD28. CD28 is a receptor that's found on mature naive T cells. And when they become activated, they will have this receptor out there to receive a co-stimulation. The co-stimulation signal comes through B7 binding it. Only dendritic cells that have antigen are going to have B7. Macrophages will express B7 as well as B cells, but not unless they're activated through an ongoing infection. So the 1-2 punch here, T cell has to recognize antigen along with CD4 recognizing MHC class 2, and the co-stimulatory signal has to be sent through B7 down, or through CD28 by B7 binding down through the T cell in order to fully get it to become activated. Okay, and this is kind of what I said. Only dendritic cells, macrophages, and B cells are able to express B7, but it's only going to happen during the presence of a true infection, which is really good because we don't want to have co-stimulation occurring when there's really not an infection and going around and stimulating all these T cells willy-nilly. There has to be a true infection for this co-stimulatory molecule to be expressed. And so B cells are not going to contribute to T cell activation. Macrophages really don't either, but it's dendritic cells that will have that initial co-stimulatory molecule. So then when now it's not even a naive T cell anymore. Now we have a mature T cell that has been activated, will now go on through a process of proliferation and differentiation. When those effector T cells that come out of this proliferation and differentiation go out to drive the immune response, they will interact with antigen-specific B cells, and then at that time, B cells will express B7 and interact with T cells, but only then will B cells be able to express B7. So they're not involved in the initial activation, but rather it's dendritic cells. Okay, but there's more. So yes, co-stimulant, there's a 1-2 punch. The activation of the T cell receptor in MHC class 2 or 1, and then the co-stimulatory molecule. But that can't just run out of control. And so there is another molecule that binds to B7, and that's called CTLA4. CTLA4 is found on T cells, on activated T cells. And it's kind of like a brake. Like if the car's going and you put the brake on to slow it down, that's what's happening here. An activated T cell is going to have a brake out there to say, hey, no, we got enough activation going on. I know dendritic cell, you're still presenting this antigen, and I know you want to keep this going, but we got enough T cells. They know what they're doing. So then CTLA4 is there to bind to B7 and kind of shut down proliferation. And so there's no more activation of T cells because there's already a very productive active clone of effector cells out there. So CTLA4 acts like a brake on the immune system. Okay, now ignore that diagram for a second here. Let's just look at the right-hand side where it says activation. And look at the top and the bottom. We have at the top a naive T cell recognizes specific antigen in conjunction with MHC molecule on the surface of a dendritic cell. That's going to initiate a crazy complex set of pathways that will turn on transcription factors and tell the T cell to do all of these things and make all of these enzymes and make all of these proteins. That's not important for us. So I'm going to cover that up so that you're not distracted by it, and just know that a whole lot of magic happens. Okay, when you get to a lab where you're doing specific cascade research or protein research on any of these, you can learn them more in depth. If you go to grad school, yep, then you can learn those in depth if you need. If you're very interested, you can read your text. It has a lot more in there, or you can read papers about it. But what I need for us, because this is an introductory course, the T cell becomes activated by dendritic cells, and that's going to turn on transcription factors that allow the cell to proliferate and differentiate into a variety of different effector cells. All of this talk between the dendritic cell and the T cell is going to happen through that synapse. So where that T cell is connected to the dendritic cell via antigen, MHC, and co-stimulatory molecule, that synapse is going to allow for this signaling pathway to occur through the T cell and tell the T cell what it needs to do now to become fully activated and fight the infection. But we should talk about one gene that gets turned on because it's vitally important to the rest of the stuff that happens, and that is the IL-2, or the interleukin-2 gene. So there will be a turning on of IL-2 gene to create interleukin-2. Interleukin-2 is a cytokine that can act in two ways. It can act in paracrine fashion on surrounding cells. It can act in autocrine fashion on itself and further activate itself. So it is a central cytokine to the driving of any immune response. Because it is so important to immune function, it is a target for any type of pharmaceutical that is used to calm the immune system down. So for example, cyclosporine, tacrolimus, those are drugs that are given to individuals after an organ transplant, and they're used to really dampen the immune system so that there's not organ rejection or tissue rejection. Downside is the entire immune system goes down, not just against that organ. So on binding interleukin-2, the T cell is going to be triggered then to go through proliferation and differentiation. And then those cells can produce, thousands of cells that are all going to have the same antigen specificity. And they're going to be just this army of cells that can go out and fight that one single specific pathogen that is causing the illness, whether it's a virus or a bacteria. So they're all going to have the same specificity to go out and fight that one, or at least drive the immune response to fight that. If it's a CD8 positive T cell, then yeah, it's going to go out and is going to fight. They're just going to look for that one virus and kill any cells that have that virus in them. Sorry, it's a long lecture. Okay. In the naive T cell population, even though a testing was done of the T cell receptors to make sure that there's not any autoreactive T cell receptors, it's not 100% perfect. And so every now and then there might be an a mature yet naive T cell that has the potential to recognize self. It's another good reason that CD28 has to be engaged in order for the T cell to be turned on, because Antigen presenting cells like dendritic cells are not going to express B7 if there's not an infection. And so if a T cell does bind antigen that is specific in association with MHC, whatever, if there's not a co-stimulatory signal that's sent, then the T cell doesn't go on and replicate. And so B7 is not going to be there if there's not truly an infection. So it's another way to prevent autoimmune infections or autoimmunity. And then also interleukin-2 won't be created. And so it just kind of shuts everything down. So it's pretty great that those two signals are needed in order to fully activate T cells. We've been talking a lot about, the process of them. We are going to dive into the effector functions in more detail in the second lecture here. But let's go ahead and just introduce them here, and then we'll talk about them again in the next lecture. But effector CD4 positive T cells, so these are going to be any of the CD4s that recognized antigen in association with the MHC class 2, can go on and they're called helper T cells. We learned that, right? CD4 positive T cells are helper T cells. And they have the name helper T cell because they're not actually the cell that's out doing the work. They're the cells that are helping all the other cells of the body do the work to achieve the goal of clearing the body of whatever's the pathogen. And so there's a whole set of helper cells, and they all have a slightly different function. And so the tissue of origin where the dendritic cell comes from, the type of pathogen that is invading, and whatever innate immune response came about because of the initial infection is all going to drive the differentiation into the appropriate T helper response. And this is all done through cytokines, and we'll look at the different cytokines that drive these. But they're very different. They have different cytokines that drive them. They have different cytokines that they produce. They interact with different cells. They have different functions, and they have different cell markers. So a lot of heterogeneity across these different T helper cells. You can see we have them listed there: helper 1, helper 17, helper 2, T follicular helper cells, and then the Tregs are a little bit different. We talked about those slightly, but anyway, we will look at those in more depth in the next chapter or in the next lecture. Now, CD8 positive T cells are different in that they only have one effector function, and that's to kill. That is their name. They're cytotoxic lymphocytes because they are toxic to other cells. And so they interact, though, with a wide variety of target cells and just kill them. They're going to kill the endogenous type pathogens, so viruses or maybe like an intracellular bacteria. But virtually any cell that can be infected by a pathogen can be killed by a cytotoxic lymphocyte. So they can interact with a lot of different types of cells. Now, because they are, pretty destructive, a CD8 positive T cell really has the potential to do a lot of damage to not only tissue that is harmed or infected, but there could be collateral damage that occurs to surrounding tissue that maybe isn't infected. And so actually there's a stronger, more stringent requirement on activating CD8 positive T cells than there are activating CD4 positive T cells. The body wants to ensure that if a CD8 positive T cell is being activated, it is because there truly is an infection that needs a CD8 positive T cell response. So they want to make, the body wants to make sure that the that the benefit that the CD8 positive T cell provides isn't outweighed by too much destruction. So what we have then happening with a naive CD8 positive T cell is going to, yes, have to recognize specific antigen in conjunction with MHC class 1, by dendritic cells. That's still true. There's still going to have to be that co-stimulatory signal with CD28 binding B7. So all of that is still the same. But then there's usually going to be some IL-2 that is being produced by the T cell to then go back and engage in an autocrine fashion to get the CD8 positive T cell to truly go out and be a killer cell. But sometimes it's not the CD8 positive T cell making its own. Sometimes dendritic cells and CD8 positive T cells need the help of CD4 positive T cells or T helper cells to produce that IL-2 to jumpstart the CD8 positive T cell to go out and kill. So the dendritic cell has to interact simultaneously now with not only CD8 positive T cell, but also the CD4 positive T cell. Engage both of those. Because they have both MHC class 1 and MHC class 2 on their surface, they're able to do that. The CD4 positive T cell is activated to become a helper T cell like it should, and its effector function then is to produce IL-2. The CD8 positive T cell's function is to become a killer cell. It's getting the co-stimulatory molecule and it's getting IL-2 from the helper cell. So with the combination from the IL-2 receptor being engaged, the T cell receptor, CD8 as well as CD28 all being connected, that will allow enough of a positive signal to say, you know what, CD8 positive T cell, you go out. It's your time to shine. So it's just a little bit more stringent, but that's really to cut down on collateral damage of healthy tissue. So with that, we'll end this lecture and we'll look at effector functions in a little bit more detail in the next one.